Metformin remains the undisputed first-line pharmacological therapy for type 2 diabetes mellitus, but clinical data reveals a persistent limitation. Monotherapy frequently fails to maintain long-term glycemic control as insulin resistance progresses. When patients hit this clinical plateau, standard practice often dictates dose escalation. That approach quickly hits a physiological wall. Pushing metformin doses higher triggers severe, dose-dependent gastrointestinal toxicity, forcing physicians to cap the treatment before optimal blood glucose targets are reached. To solve this bottleneck, researchers designed a 12-week randomized controlled trial in 2026 to evaluate a specific isoquinoline alkaloid, berberine, as a pharmacological adjunct. This line graph plots the HOMA-IR data from that trial, directly measuring insulin resistance. Over 12 weeks, the placebo baseline held flat at around 4.5. The berberine arm diverged entirely, plummeting by 1.6 points, down to 3.02. This 1.6-point drop in HOMA-IR represents a quantitative shift in insulin sensitivity. The data suggests that berberine doesn't just lower blood sugar through a secondary pathway, but actively modulates the underlying metabolic dysfunction metformin monotherapy often leaves unaddressed. HOMA-IR provides the baseline context, but a broader 2025 trial reveals how this combination impacts the entire glycemic profile. Researchers compared 12 weeks of metformin monotherapy directly against a berberine-metformin combination therapy. Translating the 2025 data, the combination therapy achieved a 38.2 mg per deciliter fasting plasma glucose reduction and dropped HbA1c by 1.2%. This impact extended to lipid metabolism, where total cholesterol, LDL-C, and triglycerides dropped significantly more than the control. Returning to the 2026 data, the combination also yielded a statistically significant reduction in high-sensitivity C-Reactive Protein, or hs-CRP, marking a clear suppression of systemic inflammation. The simultaneous correction of hyperglycemia, atherogenic dyslipidemia, and systemic inflammation suggests a broad metabolic impact. Understanding how two oral agents achieve these results requires mapping their specific molecular interactions. We established the mechanism by first mapping the baseline energy homeostasis pathways targeted by metformin. Looking at this schematic of a cell, we see metformin cross the plasma membrane and enter the mitochondrion. Its primary action occurs right here, inhibiting complex 1 in the electron transport chain. This creates an immediate metabolic bottleneck. As ATP production falls, the intracellular concentration of AMP rises. This shifting AMP to ATP ratio serves as the biochemical trigger for the phosphorylation and direct activation of AMPK. Once activated, AMPK functions as a central metabolic regulator. Its primary downstream effect is the suppression of hepatic gluconeogenic pathways, effectively signaling the liver to halt the production of excess glucose. Activating this regulatory pathway is highly effective at lowering blood glucose. However, relying entirely on this single mechanism requires higher drug concentrations to maintain control, leading directly to the clinical plateau seen in monotherapy. Berberine resolves this bottleneck by bringing its own molecular mechanisms into the cell, complementing the metformin baseline. This diagram maps berberine's independent cellular pathways. Like metformin, it actively drives AMPK activation. The two agents act in concert, accelerating GLUT4 translocation and pulling glucose out of the blood and into the tissue. But berberine deploys a second, distinct mechanism. It actively prevents IRS1 serine phosphorylation. This directly boosts the PI3K-AKT insulin receptor signaling pathway, sensitizing the cell to insulin in a way metformin cannot. Simultaneously, berberine inhibits NF-kappa B signaling. This intercepts the production of pro-inflammatory cytokines, directly driving the reduction in hs-CRP observed in the clinical data. The combination works because it utilizes dual pathways. It enhances the activity of the primary energy sensor, AMPK, while simultaneously recruiting anti-inflammatory and insulin-sensitizing mechanisms, specifically PI3K-AKT, that metformin cannot access alone. For prescribing clinicians, combining two potent metabolic agents usually raises an immediate red flag: compounding gastrointestinal toxicity. This chart tracks adverse effects during the 12-week trial. The data reveals a counterintuitive safety profile regarding common GI complaints, nausea and diarrhea. The combination group, in green, experienced lower frequencies of these side effects than patients taking metformin alone, in blue. The mechanism driving this safety profile is dose-sparing. Because berberine and metformin synergize at the molecular level, physicians can achieve glycemic targets using a lower effective localized concentration of metformin in the gut. The protocol achieves systemic efficacy while actively mitigating localized gut toxicity. This removes the primary physiological bottleneck that often forces patients to abandon standard type 2 diabetes treatments. Zooming out from the cellular receptors, the combination therapy executes a multi-organ correction. It suppresses hepatic gluconeogenesis in the liver, maximizes glucose uptake in the skeletal muscle, and modulates the microbiome within the gut. The data validates improvement across 3 critical axes: stringent glycemic control, corrected lipid profiles, and high patient tolerability. Clinically, this positions the dual therapy as a highly effective bridge. For patients failing to maintain control on metformin alone, this combination restores metabolic regulation before requiring escalation to highly expensive GLP-1 receptor agonists or exogenous insulin therapies. The Berberine metformin protocol provides a rigorously validated, highly accessible framework. It attacks the multifactorial pathogenesis of type 2 diabetes at its molecular root, amplifying efficacy while sparing the patient.